Yeah. Okay. Just got the message that it's being recorded. So, this is quite informal. Feel free to interject. You have questions. Happy to just take them as they come. I think I can't see the chat, but when I'm sharing my screens easily, but I think Chuck is going to catch any questions, or if he sees raised hands, he's going to shout out and stop me. But thank you for coming, and thank you for inviting me, Chuck and Jeff. It's definitely a pleasure to talk about some of the experiences we had, or I have had, to going through this whole translational process. So just to get started, I wanted to start off talking about sort of the traditional tenure track roadmap, and what I even in biomedical engineering, where we have lots of professors that are engaged in research that can translate, they spend most of their time thinking about identifying really cool open problems to solve. Tends to be in the biomedical sciences and biomedical engineering. They actually conduct the research, mentor students, and so forth. They publish, and then they're doing all of this in their journey to basically solve problems, have impact, and on that journey, they get tenure, or they're at least trying to get tenure. But what's really interesting, you can see, I found this sort of little image here about tenure. The factors that come in to getting tenure are really about your research. Honestly, even though there's teaching in here, how you publish, the impact you have, the research, and conferences matter, because what people say about your research weighs heavily into the tenure process. But what's really interesting is there's nothing in there about translation. So there's really no incentive from a career progressing standpoint to try to translate your technologies. And I think what Dr. Sewardson and Dr. Montague, what they're trying to do for BME is to change this culture, try to create incentives to encourage the faculty to translate, because I think they've got some great ideas and great technologies. But what I hope to show you is that I wouldn't say it's a different roadmap, but it definitely opens up a lot of roads and pathways that you have to kind of walk through, in addition to your sort of normal responsibilities. But it can be exciting if you really are passionate about the technology that you built, and if you think it actually will have real clinical impact. So to start, I'm gonna tell you about my roadmap, but it starts with the clinical problem. So I'm gonna talk about how we started with a clinical problem, conducted the research, published the research, but then move into what happened along the way in terms of translation. So this is a clinical problem of interest. It's in epilepsy. So over 60 million people in the world suffer from epilepsy. What was surprising to me when I first learned about this is that over a third actually don't respond to drugs. They're medically refractory. Sometimes they're called drug resistant. What that means is that there's no combination of medications that can control their seizures. So essentially, they're quite limited in how they can develop and as children, and what they're able to do. If you can't control your seizures with medication, you can't get in a car and drive it. You can't jump in a pool. It's very debilitating. And these are the most severe patients, so they happen to have very high morbidity rates. However, there is a potential cure, which is if you have what's called medically refractory focal epilepsy. That is, seizures are starting in what's called the epileptogenic zone. It's a region, or a group of regions in the brain that are sort of focal, localized in one area, that are always starting the seizures. And if you're one of those patients, then what the clinicians can do is find that epileptogenic zone and treat it. They can either treat it through surgery, so they surgically remove that, or they can stimulate that area to try to suppress seizures. Both of these kinds of treatments require accurate identification of the epileptogenic zones. You need to know where to find it and to be accurate about it. But you'd be surprised that the surgical success rates are 50%. So even after they take very large portions of the brain out, 50% of the time, the patients come back a year later with seizures. Okay, so getting towards the clinical problem. Of course, the outcomes are a problem, but what leads to those poor outcomes? So let's talk about the process of how they actually find the epileptogenic zone. So it's two phases. The first phase is this. They collect non-invasive brain imaging data on the patient to try to figure out where seizures might be coming from. So they'll do PET scans. If they're able to catch a seizure, they'll do SPECT. But MRI is a big an image that they rely on heavily, which is they're looking to see if there are any lesions in the brain that might be causing the seizures. At the same time, they capture scalp EEG over several days on a patient. They put the scalp cap on, the patient stays in a hospital room for several days to weeks. Why? Because the clinical team is waiting for seizures to happen. And what they do is when they see seizures happen, they start inspecting right before a seizure which channels, which correspond to regions might be having some type of abnormal activity. And then they might be able to pinpoint from there, okay, if, it's in this vicinity, it's on the right hemisphere in the frontal areas. Now, if you're a situation where you have a lesion, so if you're a patient and you actually have a lesion on your MRI and the scalp is showing that seizures start from that area, then you might actually go straight to surgery and the localization of the epileptogenic zone is pretty much done. And they tend to have very, on the higher end of success rate, 70%, not 50, okay? But more often than not, the MRI is completely normal. And all you get from a scalp is rough vicinity, hemisphere and lobe. So then you have to go invasive and probe the brain invasively to find the epileptogenic zone. So this can involve a craniotomy. So you go in for a surgery, a portion of the skull is removed. You have to place some electrodes on the skull or drill them into the brain. Then the patient recovers from the electrode implantation and stays in a hospital room for several days to weeks. Why? Because again, the clinical team is trying to wait for seizures. And when a seizure happens, they look right before that event and say which channels, individual channels, are demonstrating some abnormal activity. For example, a high frequency oscillation or a spike in the data. It's very manual, and they're looking one channel at a time. And these are trained epileptologists, neurologists, or EEG technicians. The problem, so here's the clinical problem. So with all of that data, you would think that clinicians should be able to accurately identify and treat the epileptogenic zone. But success rates are so variable. They vary between 30 to 70%, on average 50% success, which means it's not that easy. And they're not always getting it right. So what are the challenges with this? Well, if you want to find the EZ, one of the first decisions that clinicians have to make is where to put those invasive electrodes, right? You can't cover the whole brain if you're going inside the brain. You have to pick a region to safely implant inside, okay? But if they get that region wrong, they're not going to see anything. When you're sitting here with these electrodes into your brain, patients are there for several days to weeks. That means their brain's being exposed. It's very high risk, high cost to the hospital as well. High risk to the patient. Very subjective decision making. It's so surprising if you think of precision care medicine, AI in healthcare, all the machine learning algorithms out there, all these data, and yet there's no analytical tools in this workflow, in the clinical workflow, okay? So it's still very subjective. Outcomes are variable. If you go through this invasive process of monitoring, you go through the whole process of being diagnosed and then treated, you're talking about $200,000 to get the treatment. Okay, so that was the clinical problem. So we came in and said, we want to solve this problem. So what we did is we collected data from Johns Hopkins Hospital. So we took the intracranial EEG data, so these time series coming from various patients. I think this study was done on 12 patients initially. And we said, okay, we got this. We know that looking at individual channels is not enough. So we're going to build networks. We're going to track sort of the network structure over time. So what do I mean by network? Every channel, every electrode is a node in a network, and each node generates a time series signal. And what we're going to do is we're going to compute correlations between every pair, organize them into these adjacency matrices. Here's an example here, what you see, track them throughout a seizure, and compute statistics on those adjacency matrices. So for those who might know of network-based analysis, you take data, you convert them into an adjacency matrix, which is equivalent to properties of a graph, And then they compute notions of how important is a node, centrality, or hubs within the network, or degree distribution, some summary of the network. And that's what we did. We did a version of that where we found some interesting trends. In fact, we saw that if you look at how important any node is in the network and track that centrality over a seizure, we saw that all the electrodes that came from patients who had successful outcomes and those electrodes were removed, all of those brain regions had a sort of a bell-shaped signature, that their centrality was low before the seizure, high during the seizure, and went back to being low again. So not so important to very important in the network to not so important. And that's sort of, if you dump that sort of signal into a lower dimensional space to visualize, that's like having an electrode that lands in this two-dimensional reductions, this reduced space. All the other electrodes that came from failed surgeries and so forth did not exhibit those signatures. They had different shapes, as you can see here. So we thought maybe this is a signature. This is a trend we see, that every contact that shows this centrality signature throughout a seizure, that's going to be in the EZ. Okay, and that's where it all started. And that's when Chuck came into my office and asked me, as you say, what do you have? And I said, well, we just published this study. And he really thought, okay, you've got something here. And that's when the journey began. Okay, so this is the translation roadmap. I'm going to kind of give you a high level of how we moved from that research to the FDA submission over time. Okay, so as you can see here, we published that paper. It was a nice paper. It was in the Proceedings for National Academy of Science, so pretty high impact. People were very excited in the epilepsy community. And these are the researchers involved. So Adam is my PhD student, Adam Lee, and Dr. Jorge Gonzalez Martinez was the primary clinician who helped us run that study. Okay, so when Chuck came into the office, convinced me, actually I had already filed the provisional. He came into my office, said, let's do something with that provisional. So we started talking about potentially thinking of translation, forming a company. So one of the first things he said is like, okay, what else is out there? And so this was interesting. We had to really think about, okay, well, there's no clinical tools that are being used, but what's sort of on deck, right? And that's really looking at the latest research publications and other patents that might be out there that might compete with the algorithm that we had. We filed for a full patent from the provisional. Again, with a lot of input from Chuck, we thought about market assessments. You know, is there a market? What does that mean? Okay, well, who would be the customers if we were to package this technology and call this an EEG marker of the epileptogenic zone? Okay, who would buy it? How much would you pay for it? How much would customers pay for it? Can you come up with some revenue projections with some assumptions? Well, what are the risks if you try to commercialize? Will people even adopt? Will clinicians adopt this technology? Will they even be able to pay for it? So what are the risks and what are your plans to mitigate? Because if we're going to go for trying to raise money to try to form a company, you need to have the answers to all these pieces. And we did it. And he came in and said, well, okay, how did you get that curve? What did you assume? No, essentially. And it was an iterative process going back. So he basically was the soundboard of everything that we were doing at that time. And who's we? It was me, basically me. I think I was the only one at that time in the company that was invested in the technology for translation. Of course, Adam Lee, he was my PhD student. He just was continuing on doing the research and trying to get, more patients and more publications out of this. So with that said, once we got it right, we started applying, trying to get some money. And what's really interesting, I don't know if any of you are trying to translate, but when you're still in the lab, you've never done this before, you don't have to necessarily go straight to angel investor or venture capitals, VCs, right? There are actually grants available that want you to support you as a Johns Hopkins researcher in your lab to continue with this idea, this research, and move it into a sort of a phase or a position where now it can actually really be translated, right? And so these types of awards are like the JHU Coulter Partnership. So this is what Chuck was talking about, how he helped me apply for this award. Again, it ended up, I got 2 phases of this award for a total of $180K, but that money goes to my lab at Johns Hopkins to fund me and my student to further that research, right? To move it along. Similarly, there's another, MII stands for Maryland Innovation Initiative, and the state of Maryland provides funds to faculty in different Maryland universities to try to encourage them to push their research out of the lab and translate. So we had a little bit of modest funds. They allowed me to continue to do research in this area. We launched a company. So we thought, okay, let's do this. Let's do the paperwork. We launched a company at that time. I think it was called Neurologic LLC. We recruited a board of great advisors, Chuck being one of them. We have seasoned clinicians, people with business expertise, medical devices, and clinicians, epilepsy surgeons. But then, after doing quite a bit of research with the money from MII and the Coulter, we identified a limitation in our algorithm. It's not that it wasn't correct in the sense that the epileptogenic zone doesn't have the centrality signature. The problem was, to compute that, you need doctors to tell you exactly where a seizure starts in that EEG window and where a seizure ends, right? If I'm going to have a signature of the epileptogenic zone that says, okay, this is what the signature should look like before the seizure, right before seizure, then right after the onset, it should curve up and have this bell-shaped curve, then I need to know when it starts, the seizure starts and ends. But what's interesting is there's high variability between when a clinician says it starts. If you show the same EEG snapshot to 3 different clinicians, they will all point to different time points and say, I think the seizure started here. No, I think it started here. So high variability in those annotations, which makes it really difficult for the algorithm to work well and generalize, right? So it was a severe limitation that, what are we going to do? But it didn't, oh yeah?
Sorry to interrupt. There's a great question on your translation roadmap, though.
Okay.
And sorry to back up.
No
problem. The question is, you published in the proceedings, National Academy of Science, but you must have had some foresight to submit a provisional patent so that the paper didn't get in the way of the patent. Is that right? Or did
That's right. That's right. So you have to file the provisional before you publish. That's right. Yeah, but with that said, the PNAS paper actually did precede, let me be very precise, the PNAS paper did precede this particular provisional, because in the PNAS paper, we didn't argue a signature, like a marker of the epileptogenic zone. That paper just described trends and patterns that we saw in these networks and in these nodes over time. So nowhere in that paper did it propose a centrality signature. That happened upon reflection of the results. Then we thought, oh, maybe we need to think about this carefully. So actually, although let's see, this second plot here is not in this paper. It's in a follow-up, okay? But that's a very good point, which is if you have an idea, but you're also simultaneously doing the research that would disclose the information that would be in the patent, then you get the provisional out, but you can still publish, and that provisional will protect you.
Thanks, Sri. That gets at a couple of really great points. One is foresight, like think about what you're publishing if it's patentable. But the other is, there may be a distinction between what you've published and what is actually the thing that's worth patenting. It's great. Thank you so much.
Yeah, and I think for you faculty out there, I mean, you have to be careful, especially if you're on the tenure track and you're an assistant professor. One of the things that I realized, and thankfully it didn't prevent me from getting tenure, is in these years, I actually didn't publish a lot. I was just a little bit paranoid about the idea. When I say publish, I wasn't going out and giving the best talks that I could have given. Because as we'll talk about, there was a new discovery we had and I held it. And because I wanted to get it, because there's this particular field of trying to analyze intracranial EEG recordings to localize epileptogenic zone. If you Google those keywords, you'll get over 20,000 papers. So the community is out there. People are hungry for something. So it was a gamble. I mean, I wasn't putting out my best foot in this area. You know, I was giving talks in other fields because my research area is broader than just epilepsy. But I think it took a hit in terms of my publication metrics, you know, all the things that we get evaluated on. So it's a trade-off. I think any faculty who wants to go through this procedure, and you'll see all the stuff that has happened since, you know, it keeps going. I'll go through this roadmap. It's non-trivial. It's time-consuming. It's an investment. And I really hope that we change how we evaluate our assistant professors. I think translation is, you gain so many skills going through this process that you won't get out of experience. You won't gain that experience just by, you know, doing research and publishing your research. I think it's very valuable. I think it translates into how effective of a communicator you are, which means translates how effective you teach our undergrads and graduate students. Anyways, I'm digressing, but we can tackle that at the end.
Just a quick point on that is there is a Tech Ventures here is the one who will support you for that. I mean, they do the filing. You don't have to, you work with them. If you've got an idea, reach out to them early, and you have to do it well before. They can't turn things around quickly. So they're out there to help you with this. So this is that is how this happens. And
That's
it was
right.
Tech Transfer who sent me to Dr. Sarma because they knew of her technology.
Right. So actually, it's a good point. There was not a lot of my time invested in this filing, nor in writing those grants. I mean, I wrote something, but Chuck was there and you know, to help me and iterate with me and edit my grants, because you don't get that luxury when you are applying for just pure research grants at the NIH or the NSF. You know, you'll be lucky if you have a colleague actually sit down and read and edit for you. They just don't have the time. So these, you know, getting those research grants was actually a lot easier than getting research grants for just pure research. So with that said, we thought, okay, so we came up to that limitation, but then we had a new discovery. So obviously my lab is still going, right? Adam is still working. He was still at the bench and still thinking about what's the next generation. And we found something new. And it was completely independent of clinical annotations. We didn't need anything from the clinicians except give us a snapshot of the EEG of the brain, intracranial EEG, and we'll tell you where seizures start. Okay, so it was really nice because it overcame the limitations of the original algorithm. And in particular, it's a new marker for the epileptogenic zone, which we call neural fragility. If we have time, I'll go into it. But the idea is if you're fragile in the brain, if a small little trigger will cause a seizure. If I perturb your node just a little bit and I get a seizure, you are a fragile node. And then we figured out how to compute that nodal fragility on every node from the intracranial EEG. So with that new discovery, we applied for what's called a Small Business and Innovation Research Award, phase 1, from the NSF here. So one thing that's also, I don't know how, if everybody knows this, is you can also, so these awards I talked about were money that comes into your research lab to Johns Hopkins University, okay? But there is sort of free money that comes from government agencies that goes to your company, okay? This is just their way of trying to build small businesses and support small businesses. And so that's one of these mechanisms is the SBIR, and they go through phases. So they give a small chunk of money for phase 1, and phase 1 is defined as really kind of, I would call it proof of concept of your idea, of your technology. And then phase 2 is really about commercialization, getting towards commercialization. How can you move there? And then they give more money for that. And then once you're in phase 3, you should be kind of ready to roll without their support. But it's non-dilutable funds. It's really an award. And what comes with those awards are, expectations. And they also, we'll get into this, but they don't just give you the money and walk away. They give you the money, but then you go through some boot camps and training which help you, you and your company succeed at the end of the day.
Sri, there's a question backing up, thinking about your pathway here. They ask, how do you go about recruiting a board of advisors?
Okay. So it starts with, so if it's a clinical problem, so the first people I recruited were my research collaborators. So at Johns Hopkins, it was Dr. Nathan Crone, and Dr. Stan Anderson. And Dr. Anderson joined, and he continues to serve on the board. Dr. Crone kind of had a conflict of interest. He has his own company and so forth, so he's not there. But these are people that I've collaborated on these types of problems as well as other problems. So collaborators that are relevant and have the relevant expertise. Business. So in my case, I actually, when we applied for the MII Phase 1 award and the Coultership Award, through Stan Anderson, one of my advisors, I was connected to the CEO of, at that time, it was called BlackRock Neuromed. So it was a company that made the electrodes. Okay, so they developed the hardware. And he had a connection to the CEO. And so I got to know the CEO over time. He actually called in when I was presenting to the Phase 1 MII board before the decision was made. So that was, I think, really helpful. And because he was excited about the technology, he agreed to serve on the board of advisors. My husband also happens to be sort of in the corporate world, so I engaged him. And Tech Ventures introduced me to Mike Weitzfeld, who used to be head of cardiology, has tremendous experience in clinical trials, medical devices, and so forth. So it kind of starts with your connections and your connections making further connections. But certainly with these awards, you get visibility, right? So when you apply for these awards, you start meeting the boards of these foundations and associations, you make the connections, and then you can start thinking about recruiting them if they get really excited about your idea or your technology. Anything else, Chuck?
Nope, not yet.
Okay. All right, so with the SBIR, we went full force to do a huge retrospective study on neurofragility, the new idea. We started product development, so because we had some money, we hired a consultant who was a very seasoned software engineer to build the actual product that could basically integrate into the clinical workflow into the hospital. Now, when I was saying this was a National Science Foundation award, they don't just give you the 225K, they say, we'll give you the 225K, but the first 2 months you have to engage in what's called customer discovery. And this is really interesting, and I don't think companies do it all the time if they're sort of the first time they've ever started a company, but it's an incredibly valuable exercise, which is, they say get out of the building, get out there. You have a technology, you think that it's needed by clinicians, but you really don't. You need to go out there and talk to all your stakeholders. And they demanded to have at least, I think, 30 interviews that we had to conduct from various stakeholders, and we were coached through the process. We had to present our results and so forth. And what was really interesting about our customer discovery, we found we almost doubled our market size through that process. So what do I mean by that? Well, we kept talking about this technology that can localize epileptogenic zone from intracranial recordings. And then they were saying, well, I mean, can it do anything on the scalp EEG? Because we don't always know where to put the electrodes. Can you help us guide those electrodes? We didn't think that was a problem. We thought it has to be straightforward to know roughly what area to cover, but actually, one of the clinicians says it's much of an art as it is a science. It's not that straightforward, Sri. And all of a sudden I thought, wow, maybe we can apply this technology to scalp. Everybody that walks in gets scalp. Half of them get the intracranial. So we just doubled the use there. That would have never happened had we not conducted all those interviews. So it's a good exercise. We had recruited a part-time CEO. In our case.
Before recruiting, another question back on the advisors.
Yeah.
You always have to pay a salary when you recruit advisors.
No, so, we didn't. We would give just a marginal amount of equity. We're talking about 0.5%, because the time commitment is not very much. I think we were asking for no more than 4 hours a month but we typically don't even tap into them. It depends. Some advisors we tap into much more than other advisors, but on average 4 hours a month, and we just give out a marginal equity.
Good.
Okay. One of those advisors, the former CEO of BlackRock Neuromed, we recruited him to be part-time CEO. I needed help. I mean, I was kind of playing that role at this point. I took a year off, a year-long sabbatical to work on this SBIR phase 1, but I knew I couldn't sustain. And so we were able to get him part-time. Why part-time? Because he had already committed to another startup company, but was really interested in helping us out. Since then, we filed a new patent on fragility, and we filed in the United States, Japan, and Europe, given sort of the market assessment. So that's sort of in progress. Since then, in the last year, with the amount of people we've had, we hired FDA consultants. So we did have some funds to hire some seasoned FDA consultants, and basically Adam Lee and one other engineer that we hired submitted. So we submitted for a 510K clearance back in July. So we're hoping things go smoothly, and we hope to hear early in 2021. And since then, we recruited full-time CEOs. So Andy, or the CEO, the part-time CEO, he couldn't commit, and I realized, and what catalyzed this was we were being approached by a large company for acquisition, and they wanted to buy us really cheap. And my worry about that was not so much about the money, but my worry is that we have this technology. I want to see it get into the clinic. I think it works. I think what we have is really nice, and hopefully I'll have time to show you what we have. But I think it works. I think it will improve outcomes. And my worry was that if we sell to this company was acquiring all kinds of technologies. I was worried that our product would potentially go down the list of priorities and potentially never get translated. So I was just feeling the pressure of this, and as luck happened, I went to the annual meeting for this Epilepsy Society annual meeting, where all the neurologists and neurosurgeons that are involved in these procedures attend. Every single one around the world attend. There are not many of them. And I was there. I was speaking to a colleague at Columbia Med, and the lead neurosurgeon there, and showing him the fragility, because I was interested in seeing if Columbia wanted to test out the technology. And he said to me, just mentioned, you should talk to my brother. He is a medical device person. He can help you out, I'm sure, this and that. Long story short, I met him, and he happened to be in between. His name is Chaz McCann. And he got very excited about the technology and joined us. Now, what's interesting is there's a real strong connection to Hopkins here, because Guy McCann is his father, and Guy McCann was the founder of the neurology department at Johns Hopkins. So Chaz and Guy McCann has a son named Guy McCann, who's the surgeon at Columbia. But Chaz was the only one that went towards the business. I think everybody went into medicine, but Chaz, who's our CEO, went into business. So now we have somebody full-time doing everything, making this happen. We recruited a full-time software engineer. Within 2 months, we've raised $300,000 with Chaz through a friends and family round. And this is just to get us through some key milestones over the next 6 months. We applied for a phase 2 SBIR, asking for $2.7 million from the NIH. Of course, we don't know if we're going to get it, but that's going to take us through what we call phase 1 of pilots, where we're going to launch in 3 big epilepsy centers. So that's where we are today. We are in the process of we've actually submitted 3 IRBs in each of these 3 centers. We've been talking to Hopkins as a possible 4th center, and we're basically ready to test the pilot. So what I had next so I'll pause here for questions. What I have.
There's one on the chat. Does Hopkins Tech Ventures assist with market assessment and the logistics of launching a company?
Don't know. Tech Ventures has never engaged me with that, so I don't know if they provide those services. I mean, maybe you would know, Chuck, but I have not had assistance in that from Tech Ventures.
since they've opened Fast Forward and the incubator, they have moved in that direction, and they do have assistance.
Okay.
They have legal assistance for creating a company.
Okay.
And when you file a disclosure, they will often do a market assessment on that, so you can get some information there for that.
Okay. Good to know. Yeah, I think, I don't know when did Fast Forward kind of launch?
It was
A few years ago.
2015 maybe.
Oh, so it's been a while. Yeah,
Yeah.
I mean, I just didn't realize they had those services. I mean, but at that time, I think as we were getting the business people on board, I mean, they were basically taking charge of that. And you've also helped us, Chuck, with when we were applying for these grants. So
Yeah, I think that assistance from Tech Ventures scaled later. Fast Forward was early, so it wasn't all available early on.
Okay. Great. So what I was going to just give you a glimpse of what this new discovery is. So I'll pause, Chuck, that's how I was going to spend the next couple slides, was to tell them what our technology does.
Sure. The science is interesting.
Yep. Okay. So here is what our technology does. So just from a input-output standpoint, it's a software tool. Okay, so it's a piece of software that takes in a snapshot of EEG and then converts that into a really easy to interpret heat map. Okay, so the way you look at this map, if you look down the row here, each row corresponds to a specific channel on the brain. So if this is intracranial EEG, it's one of the contacts. Okay, and on the x-axis is time. And here is a snapshot where a seizure occurred during this period of the window. And red hot here means fragile node. Blue is an uninteresting node. And our hypothesis is that the more fragile, the more red, the more likely that area is part of the epileptogenic zone. And I'll get into fragility in a minute. But what's interesting about this particular heat map, and I'll show you a few examples, is that what's highlighted here in this box, and even here, I don't know if you can see this red labels, those are the channels that the clinicians annotated as epileptogenic zone. And in this example, we have a high agreement between what the clinicians thought and what we're seeing on the fragility map. And actually this was the snapshot of a successful patient. So this patient actually had successful surgery. But what I want to say to you is that this heat map is not just simply displaying the data. There's actual analytics happening under the hood, okay? So the first thing that EZTrack does is it takes the window and from that EEG builds a network model, a dynamical systems network model of the brain that says, how are each of these nodes interacting dynamically over time with each other? And then based on that model, we can ask questions. If I perturb this, it's like a virtual environment now. It's like a virtual brain. And now I can do little tests to say, okay, if I perturb this part of the brain just a little bit, do I cause a seizure? How fragile is it? And so we can take the model and we have theory that shows how to compute fragility of every node. The more fragile, the more likely to trigger seizures, the more red hot you are on this map. So I'm going to show you some examples, more examples. So this is a patient that was treated at the Cleveland Clinic, had a successful surgical outcome. What you see on the left is what's called the implantation map. It's where the depth electrodes in this case were implanted. So each of these row of dots correspond to contacts on a single depth electrode. And what's highlighted here, L, these first mesial contacts here, are where the clinicians felt was epileptogenic, okay? And here is a snapshot of EEG that went through EZTrack. Again, it's a little faint, but these channels that are highlighted in red, circled here, are what the clinicians annotated here as epileptogenic based on their standard visual analysis, their standard of care. And EZTrack has high agreement with the clinician, and this was a successful case. Here's a failed case. So here's a situation where a patient was treated at Cleveland Clinic. The clinician saw something based on standard analysis that there was something epileptogenic in this vicinity, okay? But then if you looked at the heat map, we saw some fragility in those channels, but this is a small window of seizure here. We see fragility in other channels. And more importantly, we see fragility way before a seizure in this area of the brain. So there's complete disagreement here between the fragility map and the clinician, and this was a failed outcome. Now, what's tricky about algorithms in this particular clinical problem is we have no ground truth. We have no idea where the epileptogenic zone is. So when you're trying to come up with a marker, you have no ground truth labels. So the way you do it, the way we approach this problem is saying, okay, a good marker is going to agree with the clinicians for successful outcomes and disagree with clinicians for failed outcomes, okay? So that degree of agreement should be able to predict the outcome. Another example of a failed case. So here's a case where this is where the clinicians identified was epileptogenic, but we see fragility almost everywhere here. It's a very yellow-orange map. And that could mean that this is an untreatable patient, multifoci, okay? Or it could mean that the electrodes were not optimally placed, because fragility is a relative measure, relative from node to node. So if I don't cover the epileptogenic zone optimally, then everything looks comparably fragile, okay? Again, this would just be an alert to the clinician. And one of the things we're saying is we're not telling clinicians to just read the map and do the surgery. We're telling clinicians, read the map, and if it looks like it's disagreeing with you, go back and take a second look, okay? Because we know, I mean, if you think about this, this map is just based on a small window of EEG. But if you remember the first couple of slides when I defined the clinical problem, they're collecting a lot of information on the patient. You know, MEG possibly, MRI, PET scans, SPECT scans, medical history, you know, intracranially. They have all this information to make a decision, and they make it collectively as a team. So in no way are we saying, throw all that away and look at our map. We're saying we think our map carries independent information from everything else, add it to your pool of information, and maybe you'll make a better decision, maybe you'll have better outcomes. Okay. So then last but not least, we did a full, so with the SBIR phase 1 money, we did a full retrospective study. We applied this technology on 91 patients, treating over 300 seizures across 5 centers. Sorry, we were processing 300 seizures, and these patients were treated across 5 leading centers, including Hopkins. I put this up there with Chuck's picture because he loves these pictures.
Thank you.
It makes Chuck smile, so I put them in there. All these pictures are exactly the same examples I showed you, but on a still image, right? So one thing you can do is you can take a fragility map and summarize fragility over the brain. And these are summaries. And what's nice about this, these are 2 different patients. Both of them had successful outcomes. And what's nice about this is if you look at what's outlined in black, these are the regions that the clinicians thought were epileptogenic based on their standard care, and there's complete agreement between fragile and what the clinicians thought. And these were successful patients. These are 3, so 1, 2, 3 patients that failed. In these 2 examples, there was fragility outside where the clinicians thought were epileptogenic. So this could have been mislocalization. Here was the case I showed that everything looks fragile, could be untreatable, could be, you know, not great implantation. Okay, when we use this,
there was a question about the centers that you did. How'd you get the centers on board, and what went into getting the retrospective study set up?
Right. So what I found most, well, first of all, many of the centers I was already collaborating with on research over the years. Okay, so it came with collaborations that some of them were already existing. So Cleveland Clinic and Hopkins and NIH, I already had relationships with, just for research collaborators. And so when you have, you know, longstanding successful collaborations, I mean, that's kind of one of the perks. If I say, oh, hey, I have something new, would you be willing to test it out? Chances are they'll say yes. So those are these 3 centers, okay? University of Maryland Medical Center came through with the money at the end of the day. I got the MII award, but part of the MII award was you'd get more money. My lab would get more money for the research if I brought in another institute, another PI from another Maryland institution. And so I was introduced to the neurologist at the University of Maryland Medical Center. I said, hey, I would love to work, process your data, include it in our research study, and here's some money to support some of your staff. And we made it happen. And she was included, and so were her patients. University of Miami, the lead neurosurgeon there was in my PhD lab. So we had took quite different paths, but happened to have a connection there. With that said, let's say you don't have existing collaborations, okay? What I found that gets clinicians excited and on board is if they're really part of a research study that's going to end up in a publication, okay? They are very busy. The clinicians that are interested in research, that are doing research, are typically very busy, okay? And so if they may not have the time to go to weekly meetings, they may not have the time to even write much of your grants, but they do want to be involved in research, and they actually provide very timely, important input that you should incorporate into your studies, right? So they're very valuable. You don't need them all the time, but they want to be involved. So as long as you engage them, include them as part of the research team, include them as co-authors in papers, then they are more likely to come on board. So, immediately what was really funny about both of these centers is we would get a couple of patients, we process them, and then both of them independently sent us emails saying, here's an abstract that we wrote up based on that one patient that you processed. Can we present it at this meeting? And we're like, sure. So that's how excited they are. They want to work on new technologies. I feel that the neurospace and neurosurgeons are very early adapters, like they are really into gadgets for some reason. Maybe that's part of being correlated to wanting to be a surgeon. But they're very happy to be on sort of the edge of research, right? So if you're an engineer and you're coming and you have this background that brings something new to the field, they get really excited. Is that answer the question? Yeah. Okay, so when we did this retrospective study, we used that degree of agreement to predict outcome. And basically what this is you're a receiver operating curve. You got true positive rate on this axis, false positive rate on this axis. And at the end of the day, we were able to get 73% accuracy in predicting outcome. So why is that good, we think? Well, these were 91 patients. And out of the 91 patients, 44 had successful outcomes. That's a 48% success rate in this pool of 91. So if we assume that the clinicians predicted success in everyone they treated, then their prediction accuracy was 48%. We took just snapshots of EEG to make a prediction of outcome, and we were able to do it at 73%. So this is just an indication, what we think is a strong indication, that there's value in these heat maps. I'm not going to go into details of this. We compare, but what's important about this is not only did we just validate fragility, we compared it to 20 of the most popular features that were proposed in those 20,000 papers that I mentioned, right? We took the most popular and we benchmarked fragility. When it came to area under the curve, so area under the curve is area under this blue curve. The higher that area, the more sort of discriminatory power that feature has. And fragility outperforms everything. If you think of positive predictive value, precision, fragility outperforms. Now, of course, I should have put a picture of Chuck here. So Chuck looks at this. He just recently saw this result, and he looks at this and he says, well, but Sri, beta's not so bad, right? Beta power is the second best guy. And he's right. It's not that bad, okay? But here's the problem. It's not interpretable, so it's not translatable. So here's the thing. When we did these predictions, they were based on random forest classifiers. So took these sort of sophisticated models from the machine learning community to do the prediction. Now, a random forest classifier can actually discriminate between 0.000015 and 0.000016. But the human eye can't. So if we were to display a fragility heat map, this is what you'd get for an example. And if you take that same EEG snapshot and computed beta power, the second best feature, this is the map you'd get for the same window. You zoom in on the regions that popped out of the map as red hot, very fragile. You see it very clearly. there's a contrast between a sea of blue and green with the red. But if you look at beta power, it's not so clear, especially here. This was bright red here. I'm seeing nothing compared to the rest of this background in the beta map. So how is this map going to help a clinician? So then we computed a contrast ratio, what we call an interpretability ratio, and we saw that fragility was much more, significantly more interpretable than all those other features, okay? And this is really important. When you come up with an idea, it has to be translatable. It has to be adopted, which means it has to be, you have to kind of have all of these features in there, not just predictive power. They're not going to be happy with a black box where you stick in an EEG and say, oh, that guy's going to fail. That's not going to help the clinician. You have to give them information.
Here's a tough question along that line. At this stage, what if fragility wasn't the top performing metric or feture? How would you proceed?
Oh, well, a little bit of panic. Remember that emoji? I would have that reaction again. You know, I would just go back to the research board again, go back to the drawing board, understand what it is about fragility that didn't do well. Maybe I would be, you know, take the top guy and say, well, what's going on here? Has anybody tried to put this? Has anybody tried to make use of this? Maybe that's something I would pursue if it hasn't been or is not being pursued. But I would definitely go back to the drawing board. One of the things that I feel what's very different about fragility than any of these features is it's based on a hypothesis of what's happening inside the brain, in an epileptic brain, that was formulated by clinicians. So based on all their experience over the years, they have an idea of what they think's happening in an epileptic brain and how that's different from a normal brain. And when they communicated that notion to me, I translated that into the mathematics. That's how we work together, okay? All of these other algorithms, you don't even have to talk to a clinician to come up with those ideas. They are just a combination of signal process. Let me process the data. Let me do spectral analysis. Let me look at the power in different frequency bands, or let me put these things into an adjacency matrix and look at network statistics, like our first approach. None of these tell you anything about why that brain is pathological, why that epileptogenic zone is epileptic. None of them. Okay, so I think that's what is a real departure between our approach and what happened here. So I wasn't surprised when I saw these results. But with that said, go back to your research lab and try again. Okay, just a quick thing. So what's nice, so one of the things that's really important if you're trying to translate a technology, a software in particular, into a hospital clinic, okay, is integration with their existing system. So we're pulling data from their clinical servers and processing it, right? So it's tricky. How do you do this safely? How do you do this that's in a HIPAA compliant way and so forth? Well, we realized that the fastest way to do this is plug in, we call it's EZTrack, plug in EZTrack into one of their existing tools that they're already using, okay, where they're already looking at the data from these tools. If you remember that picture of the clinician visually inspecting, they're sitting in front of a computer screen and they've got an application up that allows them to pull in data and look at the EEG, raw EEG, and even annotate it. Well, we plug EZTrack in there, they annotate it, they select a region of EEG, and then press a button that says generate heat map. That goes to the cloud. So we're using Amazon Web Services, goes to the cloud, builds the heat maps, and the files get saved back onto their desktop. So it's fast, it's scalable, and it's secure. AWS is HIPAA compliant. And in some sense, this got rid of sort of the messiness of trying to work with hospital IT, integrate a new tool, put in a new workstation. We got rid of all that. We actually integrated with an existing tool that already had the GUIs in place, and we said, okay, now just add this new feature called EZTrack, and then the clinicians can use it.
There was a question. Could beta data be contrast enhanced rather than eyeballing the display? I mean, I guess that's with the heat map.
Yeah, you can't scale this in any reasonable way to make it look. I know, good question. No, you can't easily do that. You would have to come up with some bizarre nonlinear mapping to make it work for this map. but then it may not work for another map, right? So that's non-trivial, right? And the beta power, not surprised that it works reasonably well, because actually one of the signatures that the clinicians look for is what they call a beta buzz. So beta is 10 to 30 hertz on average. And so the beta buzz is if they see on the individual channel, you know, squiggles that look like beta frequency, that is a signature that they look for to say this particular channel is epileptogenic. Okay. The problem is that's just an analysis on the individual channel. So beta power, although it might have useful information as seen in our analyses, it's useful. It's providing information. It's not taking into account interactions in a network. And so it might have red little blips here and there, but it's not going to give you anything consistent, transformation or not.
Well, with that, we're at the end of time. If you want to you got any last words, last thoughts here that you want to end with, we can then.
I just wanted to bring in the pictures. Yeah. I would say that it's a lot, right? So when I just show that first slide of if you're an assistant professor, it seems like it's not trivial to, come up with important problems, solve them, and publish in journals and make a name for yourself, and if you talk to any assistant professor, they will look stressed. They will say, I've got too much on my plate. And I don't blame them, because it's a high bar, especially if you're at Johns Hopkins, to meet that tenure bar. But translation is non-trivial. It's a lot of blood, sweat, and tears. And I think that you have to be super passionate about what you're trying to translate. You have to believe in it 100%. Of course, if you don't believe in it, you're not going to waste your time, but you have to believe in it. You have to believe that it's going to make lives really, really have an impact on people's lives. And you have to be willing to do those late nights and weekends and whatever it takes to make this happen. Because unfortunately, it's not used to evaluate the value of a faculty member. It's just not. So in today's environment, you have to kind of go above and beyond and take that extra time to realize these dreams. But it's fantastic. You meet a variety of really, really smart people that are not in your area, not in your field. You learn a lot about business, marketing. You wear multiple hats. I absolutely think that, I'm going to stop sharing here, that this translates into being a more effective faculty member. For example, I have to be able to communicate a technology that uses dynamical systems modeling, optimization, matrix theory, things that are complicated. And I have to be able to explain that to someone who hasn't taken math since high school. How do you do that? Well, you know what? When you're doing computational modeling like my research lab, I'm going to be submitting grants to the NIH, and there's going to be a clinician on that panel because I work on neurological disorders and diseases. That clinician hasn't taken math maybe since undergrad. So I have to know how to explain the value. I have to convince this very smart, experienced person that I've got something innovative here, that I've got something that is a game changer. You only get that. You gain those skills through a company as well, right? So all I'm saying is that if you take that translate, it's not an orthogonal sort of set of experiences. They do help you become a more effective faculty member. And in fact, if anything, now I'm in this sort of vice dean role. Vice dean is like, okay, all of a sudden now the programs are products, the students are customers, and we're trying to figure out how to increase the market. We want the School of Engineering to generate revenue. We want brand. We want higher enrollments. We want visibility. We want better rankings, right? All of this makes you a more effective administrator, if that's what you want to do. So I'll end on that.
Well, thank you, Sri. That was great.
So we're able to take more questions, but at this point, I'd love it if everybody could unmute your microphones, and please join me in thanking.
Thank you so much, Dr.
Sri Sarma
Thank
for
you.
this talk.
But just one last question before you go.
Yeah.
Oh, please, let's. We have time for a few questions, Dr.
Yeah, having a dedicated CEO. That was your approach, and is that sort of the or standard, or do you recommend that for all the clinicians, surgeons who can be quite busy from their other responsibilities?
Yeah, so we just hired our CEO 2020. So right, so this kind of journey started 2015, and we just got the full-time CEO in 2020. I just don't think we were ready to recruit someone of that caliber, right? That could actually make this happen. But there's no question, nothing can replace a seasoned CEO. Like nothing. I mean, even if I can sell my technology, everybody sees my passion, I can go in front of VCs and so forth. But at the end of the day, I didn't know anything about FDA, by the way. I don't know anything about quality management systems. I don't know, nor do I care to do or want to do revenue projections on an Excel sheet. I don't want to go out there and sell. I don't want to make all those crazy, I can't do all of that. So it was almost like a release of pressure that now I can hand my baby to someone else, and he's going to take that baby and develop it and make it grow. I mean, it was just wonderful. And so unless if you're a busy clinician, unless you have aspirations to be a CEO of a company, or you want to try do it yourself, I mean, I wouldn't necessarily recommend that if this is your first venture, like your first, company. You definitely don't underestimate the value that someone with experience can bring in as a CEO. They really know how to run a company. They know how to manage people. They know how to think about long-term, short-term strategies. They know how to plan for it. They know how to gather the resources for it. They have to identify the resources. They gather the resources. I mean, it's non-trivial. Right? And it's not that we can or cannot do it. It would take us a learning curve to do it as well as they do, and it takes time. And if you don't want to do it, then don't do it, right? I mean, but getting that quality CEO, you have to have something extremely compelling. So you want to get it to that point where you can sell it to a great CEO and recruit that person to come on board.
Thanks.
What's the minimum financial capital you need to have a CEO on your
So that is a hard question to answer.
Okay,
So
gotcha.
we were able to recruit Chaz on pure equity. Okay. So he's willing to work 6 months for no money, but he also raised it. So
Gotcha.
he says, okay, I'm going to raise it. If I can't raise the money to pay myself and to pay all of you, then we don't have anything, or I'm not good. Right. And so that's exactly what he did. So he came in on equity and happened to raise enough money. Now he's not going to take a big chunk of salary. He understands the limit, our financial constraints. And so, it just happened to be that he's in between companies. He's financially stable, so he can do something like this. So it really depends on the person. Yeah.
Just a quick note for those still around. I put a survey in the chat. If you have just 2 or 3 minutes to take that survey, it would be great on the way out. And you can leave your email for contact. It has my email if you want me to help, since Sri has been pointing out that I get involved with this.
The question of how to choose a great CEO is a really great question, and I'm recalling from a previous seminar in this series actually where we touched on that as sort of one of the elements of that's so important to translation. And I'm going to try to dig that out, Dr. Lee, but it's there are a number of really important considerations that you want to have in mind on what makes a great CEO. Should they be involved in the foundation of the company originally, which was your original question? Not necessarily. How much should you pay a CEO and things like that is great. And I'll try to dig up that information. We actually had a nice set of principles to consider.
Great.
Yeah, and I think a good sort of indicator of a good CEO is when they are thinking about joining, that they go and talk to your customers and find out what do they think. He literally went I didn't realize he was doing this, but he went to all the leading surgeons that he knew, because he has a brother in the field, right? And he asked all of them, what do you think about this team? What do you think about this technology? He did all these interviews on his own to be convinced that there's something real. Mm-hmm. And that's a good sign, right? You don't want somebody to just be sold by you. You want somebody who's going to go out there and say, this will sell, right?
If you pay a CEO too much, are they invested in the company or are they just taking it? So.
Mm-hmm.
A question in the last few seminars in this series, we've talked a lot about 510K process.
Mm-hmm.
And you're in the middle of it now. And so some basics. Was the 510K clearance that you were seeking, is it 510K? I'm guessing. Was it class 1? Was it class 2? Was there a predicate? Can you comment a little bit on just your 510K mechanics that you dealt with? What were the hard parts?
Yeah, so yes, it's a 510K. Chuck, is class 2 the minimal risk? Yeah, class 2.
No. There's class 1, but it's a class 2. 510Ks have to have a predicate, and you did have a predicate.
And we had a predicate. Yeah, and that predicate was a device that had a retrospective study based on 19 patients, and we just submitted with 91. So that's but it was a while ago. I didn't go through the process of the submission, but it was extensive. Like Adam Lee had to write all the software requirements documents and things like that. And we worked with our consultants who helped us with that. But minimally, you need to find the predicate device.
You brought in a special regulatory consultant.
That's right. Actually, Chuck, when I was with the Coulter Foundation and had that award, one of the perks of that award is we got like an hour with a seasoned FDA consultant that Chuck was part of the program. So he was with me when I had my 1 hour interview or session with her. And then immediately she asked me, okay, tell me about your device, some of that. And she brainstormed, she looked up things, she immediately found a predicate device. She said, here's what you should be doing. And that's when it started. And then I ultimately hired her company, consultants from her company, to finish the deal when I got the money to do, and when we were ready for FDA submission.
That's great, Sri. Thanks a lot. That's good to hear. It sounds like you were able to navigate that. Yeah.
Ahmed, go ahead.
I had a question. So thanks for the great talk. And so I'm a PhD student, and we had also like a new algorithm or software that we wanted to actually patent. And we talked to the patenting office back in early 2020. And so at the time they told us right now there are new rules that makes patenting softwares, if you don't have any hardware accompanied with, it's like really hard that we do not advise you to go for the patenting, and instead just do the licensing and start publishing. So I was wondering, is it like that way, or because you mentioned that you are also trying to patent new software, so did you have the same problems or new rules?
Yeah, well, I recall, and Chuck might need to clarify this, but yes, I definitely recall when we bring any ideas now to the IP people, at Hopkins, and we say it's a software, that's the first thing they'll say, that it's very difficult to just patent an algorithm, and you absolutely can't patent an algorithm that just automates what the clinicians do. So that's not patentable. But we somehow were fine. I don't know, Chuck, what's the definition of a software not being able to be patented?
Well, you can't just, no matter how complex and how long, if it's just process that the computer does, it's a technical issue. That's why we bring in legal people who know how to navigate this. And many patents have been overturned because they just, what looked like it was creative wasn't. It was just pure calculation, no creative element. There's something called a software plus, and I can't go any further than that. Might be an idea next. We'll add a patent lawyer to bring in and talk about that specifically, the challenges there. But it is something that is so complex, and it's but it really has muddied the water and does make tech ventures very cautious about patenting for good reason.
You're probably interested, Hamed, on a couple of the earlier seminars too. This summer we had software as a medical device. Check out that one. And Antigone's
on patenting software.
Yeah. they actually said, you know, the idea is good. I mean, it is possible for patenting, but it just doesn't worth it because it may take, it takes you 1 or 2 years and you won't be able to publish anything. So that was what we were told. And then they said, we just say that you go with the licensing right now and start publishing. That was what we were told.
Shri, we're going to give the last word to you, and we're 15 minutes into overtime, and so we'll wrap. Shri, did you want to respond to the last bit about software as a medical device or any other travails in the patent or 510K experience?
I would just say, I mean, definitely engage the Hopkins IP people because I've always found them extremely helpful, and they will tell you what you can and cannot do and what your options are. But don't be, I guess, disappointed if they say it's not patentable, because there are several technologies out there that are very useful, translatable, that are not patented and not patentable. So, but you do really need to understand what is and what is not patentable, and then whether you want to move forward with what you have. And maybe now with the Tech Ventures services, maybe they could be helpful in advising you guys, in terms of, okay, I can't file this patent. It's not, patentable. What do you think? Is it worth, is there a market out here? Can I actually commercialize this? I would think that they would be helpful.
Mm-hmm. Very good.
Okay.
Awesome. Thanks again, Shri.
Thank you.
Thank you. Okay.
Thanks, everybody.
Thank you, everyone.
Thanks, Ro.